Welcome everyone to this Working Group 5 video training series organized by Alex Almeida. This is part of Working Group 5, and Working Group 5 investigates the links between nutrition, the gut microbiota, and GIBA and their impact on health across the life course. I am Patricia Sanz Morales. I'm a postdoctoral researcher at the University of Reading. I finished my PhD quite recently in 2025, also at the University of Reading, and I'm happy to be co-leading Working Group 5 as an early career researcher. This presentation is going to be a whistle-stop tour on biological signal, clinical silence, translating microbiome modulation into patient benefit. So a bit of an overview of what I will be talking about. I will be using a case study. This is a clinical trial using human milk oligosaccharides in irritable bowel syndrome adult patients. The results are currently being published, so very hot off the press. I will also be giving some background evidence, in vitro gut model evidence from our lab. The clinical translation challenge, so what happens when we have a biological signal but no meaningful results in clinical results, and how to better frame these findings. It is important to still publish results that may seem non-significant, and some take-home messages right at the end. The case study I was talking about is a clinical trials registered crossover trial in adults with all subtypes of IBS. 42 people enrolled, 37 completed the study. And this was a 5-gram dose per day of a human milk oligosaccharide mix for 28 days, followed by a 28-day washout period, after which people were switched to, again, either 5 grams a day of the HMO or placebo, which was maltodextrin. And the primary outcome was self-reported, so IBS symptom severity score. Secondary outcomes included gut microbiota composition measured by 16S RNA sequencing, as well as quantification via FlowFish, so fluorescent in situ hybridization flow cytometry. We also performed urine and fecal NMR, sleep quality scores, anxiety, and depression. And this was a clinical trial in IBS using HMOs. This is what the trial looks like, so crossover with 4 visits. We took samples and recorded results before and after each of the 2 treatment periods. So why HMOs in IBS? So just to introduce HMOs as the intervention, HMOs have prebiotic potential. They are non-digestible glycans that reach the colon intact and can be selectively utilized by key microbial groups such as Bifidobacterium. The fermentation logic, so we expect there to be a downstream signal, so a shift towards a more saccharolytic metabolism with organic acids and metabolite profiles more consistent with gut homeostasis, which is a problem usually in IBS. So HMOs can shift the microbiota, shift the metabolome, and hopefully improve symptoms. That was our hypothesis. IBS patients often show altered microbiota, metabolites, and this may be linked to the typical symptoms such as bloating and abdominal pain and discomfort, which influences quality of life. Our in vitro results also suggest that HMOs increase bifidobacteria and other beneficial microbes such as Faecalibacteria prosnitzi. These are some results from a recently published study from our lab using, again, flow cytometry to tag these bacterial groups and quantify them. We compared IBS versus healthy individuals. N equals 10 in this case for this analysis. And we basically were confirming prior literature before selecting a HMO mix to take forward to the clinical trial. You can give that paper a read. And we also confirmed that concentrations of short-chain fatty acids were increasing. There are many types of HMOs which we also screened against a positive control, which was FOS. And the HMO mix number 2 outperformed the individual HMOs and the other HMO mix 1. And again, these results will be published from the clinical trial. HMO mix 2 was a 1-to-1 ratio of 3SL and LNT, which are manufactured and available commercially. So the clinical translation challenge. IBS is a biologically plausible target, but symptoms are multidimensional. It's a complex condition. As I mentioned, it's involving pain, bloating, dysregulated bowel habits. The gut microbiota also seems to be some form of dysbiosis there. And the metabolome, by consequence, also is dysregulated. This trial gives us a useful case study because it separates biological activity from symptom efficacy. So we have separated primary outcomes from secondary outcomes. And the clinical silence, the title of this short training video, comes from there being no superiority of the HMOs over the placebo in the self-reported IBS symptom severity score primary outcome. I must mention here that treatment A was the HMO and treatment B on the right-hand side was the placebo maltodextrin. So you can see very heterogeneous panel at the very bottom from the grouped scores, and no symptom benefits in the second to bottom panel from the HMO in pink, and a slight decrease, so big placebo effect, which has also been reported in the literature before. So placebo effect in IBS has been an issue in the past. Up to 60% placebo effect has been reported. And this was a similar case. However, we did see microbiome shifts. So, for example, between visits 1 and 2 on the left side, we see the HMO increasing bifidobacteria on this volcano plot, whereas practically no changes were seen with the maltodextrin on the right-hand panel at the top. There were similar case at the bottom. So the bottom is combined visits 1 and 3 and visits 2 and 4. So before intervention versus after intervention. The results are magnified. I won't go into detail here. The interpretation, however, is that symptoms did not improve beyond the comparator contextual response despite biological activity. So there was a placebo response, which was, again, typical in IBS. Clinical endpoints must therefore be interpreted alongside mechanism-linked biology. Even though the intervention was not inert, successful modulation of the gut microbiota did not equal clinical efficacy. So central questions for this training video is what do we do when a microbiome targeted intervention changes the microbiota metabolome? What else is needed for that signal to become a clinical response, which is what we would have liked to see in a clinical trial, which is testing a potential new therapy for IBS? What are the barriers and important considerations? What could have been improved here? Of course, there is always the issue of time and money, but we want to understand what are the plausible translation barriers. I have 5 examples on this slide. So why can biology shift without symptom benefit? Placebo and context, top left. So IBS outcomes are highly placebo sensitive. As I mentioned in the past, there can be up to 60% placebo effect in IBS. So we have to set our expectations correctly and expect a masking of the therapeutic benefit, comparing it to the placebo. There is also a high responder heterogeneity. So we included all IBS subtypes in this trial. This included IBS-C, IBS-D, IBS-M, and IBS-U. And all of these participants had a different baseline microbiota, as well as diet. Even though we recorded diet, we could not control for diet. We just encouraged participants to maintain their habitual diets. And their symptom drivers may determine who responds and who doesn't to the therapy. It is well known that people have different triggers for IBS, such as FODMAPs or stress and anxiety. The dose, duration, and timing, so bottom right, as I mentioned, we don't have unlimited time and money for our research as much as we would like. So the intervention may have been insufficient to see clinical benefits, even though we saw metabolome and microbiome shifts. There is also symptoms which are network outputs. So pain and bowel habit integrate motility, sensitivity, stress, diet, sleep, and immune signals. And even though we measured some of these as secondary outcomes, we did not power the study for these secondary outcomes. And bottom left here, so endpoint mismatch. Microbial shifts may affect barrier immune biology without changing IBS symptom severity score over the trial window. This does not mean that we have a completely inert or non-significant trial. So what would a stronger translation design test? So we could move from did it change the microbiome to who benefits through which pathway. So for the next person who studies this therapy option in IBS, we want to understand baseline phenotyping is important. This can lead to mechanism-led interventions such as HMOs with early biological response, which can be measured. We know this as well from our in vitro models. And we can measure mediation by analyzing it and hopefully see patient benefit. We can also recruit more participants, so increase the power of our study, which will also allow us to stratify before randomizing. So if we were to still include in future all IBS subtypes, we can stratify by IBS subtype. We measure baseline bifidobacterium, baseline saccharolytic capacity, habitual fiber intake, FODMAPs, which are also important in IBS, psychological burden, which forms part of these symptom triggers in IBS, and medication exposure. Our participants, some of which had other first-line therapies and second-line therapies, were recorded in the study. We did not, however, control for this. So we can also pre-specify mechanistic endpoints like microbiome composition, which we did, short-chain fatty acid and organic acids analysis, which we did via NMR, as well as GCMS for targeted organic acids. We can also measure barrier and immune markers, which we did not have time to do here, as well as cortisol, HRV where relevant. We can also define clinically meaningful responses. So we used IBS symptom severity score here, and we also used other validated surveys, such as the Pittsburgh Sleep Quality Index, to measure sleep quality. But we need to align timings and expected domains benefits with the biological mechanism. Practical message here is a biologically active prebiotic may need responder enrichment rather than broader dosing. So how to better frame these findings? It's important, as I mentioned earlier, to still publish what can be non-significant results, in this case, for our primary outcome. So we have to be honest with our conclusions. Instead of saying HMOs have failed in IBS, maybe say HMOs produce measurable biological modulation, but this was insufficient in this case to deliver group-level symptom superiority. We have not focused here on personalized nutrition intervention. We were hoping to be able to treat as many people as possible. That's why we included all IBS subtypes. So what does the trial actually show? It shows that HMOs can engage the gut microbial ecosystem in adults with IBS. What it does not show is a universal unstratified symptom benefit over placebo. What it enables is better responder hypotheses and mechanism-led trial design. So some take-home messages is that the value of the trial is translational. So we saw that microbiome modulation is measurable, it's possible, but it's not sufficient as a clinical endpoint. We saw that IBS trials need responder-aware design. Baseline microbiota measurements, metabolome, diet, subtype, and psychological context may determine benefits. And clinical silence can still be scientifically informative. So it tells us where translation has failed and how to design the next study more precisely. Thank you very much for listening, and I will be answering questions in our live Q&A. Stay tuned.